So now we're on step 4, production of tissue damage. There's a number of reasons a pathogen might benefit from damaging your tissues. One is to aid spread of the infection to other parts of your body. So it gets rid of the tissues that are blocking it, essentially, or bores through the tissues that are blocking it. Another reason is to eat you, because remember, these are chemoheterotrophs. They are using organic molecules for both energy and building material. Those organic molecules are coming from your body, so technically these microbes are eating you, and that's going to cause some tissue damage. Competition with the host for resources. So sometimes this is similar to eating, but sometimes there's a nuanced difference here. So maybe instead of breaking down your cells for food directly, maybe this pathogen is just taking resources from you that you would have used to build a new tissue. That prevents you from doing your own tissue repair. Maybe caused by the host's immune response. So sometimes the pathogen isn't hurting you directly. Your own immune response against that pathogen is hurting you. And that, usually your immune response is helping you, but if your immune response goes haywire, and that's the case with the Spanish flu, your own immune system can cause tissue damage. And usually tissue damage is caused by toxins produced by the microbe. There's two different kinds of toxins. There's exotoxins and endotoxins. So exo means outside, or outside. So exotoxins are secreted out into the external environment of the bacteria. So the bacteria excretes an exotoxin into your bloodstream or into your body fluids. Endo means within, so endotoxins are part of the bacteria, but it's actually very specific. So endotoxins are part of the gram-negative outer membrane. So that's a very specific thing. That's all an endotoxin is. It's a component of the gram-negative bacteria outer membrane. So let's talk about spread first. So there's a number of ways bacteria can spread through your body. So some bacteria bring kinases to dissolve clots. So we talked about how some bacteria cause clots in order to protect them. If they clot your blood around them, that creates a wall around them that protects them from your immune system. But other bacteria will bring kinases, which dissolve clots. Because when your body makes a clot, not only does it prevent you from bleeding out, but it also prevents the spread of bacteria into your body. So if you have a wound, there's a lot of bacteria in the environment that could capitalize upon that breach in your protective barrier, the breach in your skin barrier. And so they might want to enter through the wound. But you have these blood clots that prevent bacteria from entering. So blood clot, when you make blood clots, it prevents you from bleeding out, but it also prevents bacteria from getting in. If a bacteria has a kinase, it can break down your clots, and that gives it entry into your body. Also, if you already have an infection in your body, your immune system will create blood clots around the infection to contain it so it doesn't spread. But again, through the use of kinases, some bacteria can break down those clots and spread. Destroying connective tissue. So there's two enzymes that can destroy connective tissue that we'll talk about, hyaluronidase and collagenase. So connective tissue, it's just like a solid barrier. It's a wall that the bacteria have, it's not easy for them to get through or get around. So instead of trying to find a way around it, some bacteria can just bore straight through it through the use of these enzymes. Hyaluronidase breaks down the extracellular matrix. So in animal cells, the extracellular matrix is what holds all the different cells together. So the reason your cells aren't just falling apart is because they're all held to each other with an extracellular matrix. So hyaluronidase is an enzyme that some bacteria can make that breaks down the extracellular matrix so that it has an easier time moving through your body. And so that increases the permeability of tissues. Then collagenase breaks down collagen. So, and again, that just breaks down. Collagen is a part of a lot of connective tissues in your body. So bacteria that have this can just move right through them instead of going around it. All right, viruses. Remember that viruses do a lot of things that damage host tissue. So first, they're going to stop the host cell from transcribing and translating its own material. It's also going to stop the host cell from performing mitosis so that the host cell can no longer divide and replicate. And that's all to encourage self-replication and spread. So instead of allowing the host to perform transcription and translation for itself, the virus redirects all of that transcription translation equipment to itself so that it can make more viruses. And then when the virus exits the cell, it lyses the cell. It breaks it open. So that's going to damage the cell. And if a lot of cells are affected in a particular tissue, that's going to damage the tissue as a whole. All right, eating. So what do microbes need to support their own growth? So remembering back to microbial growth chapters, they need energy, carbon, nitrogen, phosphorus. And now viruses don't really have to eat. They don't really have metabolic processes so much. This is really more for bacteria. So some pathogens may specialize in catabolizing sugars, fats, proteins, maybe even specific amino acids, but whatever it's eating, it's coming from the host. It's coming from your body. Okay? So living human pathogens are chemoheterotrophs. They get nutrients and energy from the organic molecules found in your body. So by breaking down your cells, by breaking down cellular products, those are the places that they're getting, a lot of those pathogens are getting their nutrients and their building materials. So pathogen, remember, is a subcategory of parasite. So pathogen and parasite those are actually interchangeable terms. There's not a clear distinction between them. Remember, a parasite is something, it's any living organism that's living on or in another organism and that's benefiting at the expense of its host. So the microbe, in this case, the microbe is benefiting at the host's expense. The host is damaged by the relationship. That's what a parasite is. A pathogen is a kind of parasite that causes disease. Right? So all parasites are going to cause some kind of disease because they're benefiting at the expense of their host. They're causing negative consequences to their host, and that would be called disease, right? But sometimes we might say something's a strict parasite and not a pathogen if it's just taking nutrients from the host, but not causing any other symptoms other than nutrient deficiency. A pathogen might cause other kinds of symptoms aside from a nutrient deficiency, but they're really fairly interchangeable terms. So pathogen is a subcategory of parasite, which is a subcategory of predator, actually. So parasites are merely predators that live on their prey. So you are the prey for your pathogenic microbes. So parasite is a predator that lives on or in its prey. A pathogen is a parasite that causes disease symptoms rather than merely nutritional deficiency. Okay, competition. So competition is slightly different from eating because in competition, maybe the cells are not being eaten themselves, but nutrients that are flowing through the bloodstream are being absorbed instead of being used for the host. So being taken by the pathogen instead of being used for the host. So let's use iron as an example. Iron is a limiting micronutrient for microbial growth within an animal. This is because even though iron is present in decent amounts, it's usually complexed with organic or biological molecules in the host so that it's not accessible to the microbe. So when I say a limiting nutrient, means so iron is a limiting micronutrient for microbial growth within an animal. That means that the availability of iron is what's holding back microbial growth. So if iron became, you can add more sugar, you can improve the temperature, you can improve the pH. Nothing you can do will improve microbial growth until you give them more iron. It's limiting their growth. So the reason iron is limiting to microbial growth in an animal host is because even though there's lots of iron, plenty of iron around, it's complex in biological molecules already. So it's not available to the microbe. So for example, you have a lot of iron in your bloodstream, but it's all bound up in the center of hemoglobin. So here's the protein hemoglobin. It's found in your red blood cells. It binds to oxygen. Those little red centers, that's where the iron is. The iron is in the little red centers, and that's where oxygen binds. So this is the protein that moves oxygen through your body to all the tissues that need it. Now, this iron is not available for any pathogen that needs it because it's already used. It's already bound up in this larger molecule. So some microbes will come with these proteins called siderophores. Siderophores are proteins that help the pathogen extract iron from other proteins or from other molecules. And so it's basically, it's just an iron scavenging molecule. So siderophores can pick out iron that's already complexed in another organic molecule. So that would be an example of competition. It's not necessarily eating the host, it's just taking nutrients that the host needs to build all the molecules that it needs to build. Self-damage. All right, so self-damage is when, what I mean by this is when your own immune system is causing damage to your body. And we have a few examples of this. So interferons are where we'll start. Interferons are specifically your response to virus infection. So when you have a virus, your cells release interferons. Interferons inhibit RNA and protein synthesis inside the host cell and all the neighboring cells so that the virus can't use the cell to produce its own capsid and enzymes anymore. So remember, why is the virus entering the host cell? Well, what the virus needs from the host cell is to replicate its genetic material and transcribe and translate its proteins, its viral proteins, to make more virions. If transcription translation is unavailable, the virus cannot make more virions. It cannot make more viral capsids. It cannot make the enzymes that it needs to escape the cell. It can't make anything that it needs to replicate. So when your cells have a viral infection, they release interferons. The interferons shut down transcription translation in the virus-infected cell and in all the neighboring cells. Now, that's great to prevent the virus from replicating, but it also kills your own cell because your own cell needs transcription translation to live. Your cells need to make proteins to survive, right? Because proteins are they're the little robots that keep everything going, that keep life running in the cell. So this is an act of self-destruction by an infected cell to prevent spread of the infection. So obviously this is a defense mechanism that can only work for viruses because bacteria do not need to use the host cell's transcription translation equipment to proliferate. So let's look at a picture here of this. So here's a picture of interferon. So you have a virus, here's an RNA virus, like maybe COVID-19, entering the cell and uncoding. So now you have the viral RNA. The RNA needs to now be transcribed and translated into, the viral RNA needs to be transcribed and translated to make more viruses, right? You need to transcribe the RNA into new RNA to make more viral genomes. You need to translate the RNA into capsid proteins so that the virion can be put together, can be assembled. Now, once the viral RNA is in the cell can sense it through a variety of chemical pathways. Once the cell recognizes that it's been infected by a virus, the cell is going to initiate transcription translation of interferons. So here's an interferon. This interferon, once transcribed and translated, it's going to shut down transcription translation here. It's also going to be released to the neighboring cells to just shut down transcription translation here. So this is a race because if viral proteins get transcribed and translated first, not only are they going to spread to the neighboring cell before the interferons get there, but they'll also shut down transcription translation of interferons. So if the virus gets control of the cell's transcription translation machinery first, no interferons can be produced because it's no longer in control of the host cell. But if the cell detects viral infection first and makes those interferons, the virus can't replicate. And then these interferons go to neighboring cells to shut down transcription translation here. So here, transcription translation is going to be shut down here to prevent the virus from spreading. Also, antiviral proteins can be produced. Now, once interferons are produced and transcription translation shuts down, this cell will undergo apoptosis. Apoptosis is programmed cell suicide. So this cell is committing suicide in order to prevent spread of this virus to other cells. Host cell signal, okay, so going back to self-damage, host cell signals immune cells to destroy it. So sometimes a cell that has been compromised by a pathogen, like a pathogen has entered it, like a virus or an intracellular bacteria, that compromised cell, that infected cell, will signal to immune cells that it needs to be destroyed to prevent the spread of the infection. So let's look here. You have a pathogen that's entered the cell. It could be a virus, it could be a bacteria, it doesn't really matter. Now it's hiding from most of your immune system by entering the cell. So now most of your immune pathways cannot detect it. What this cell is going to do is it's going to present molecules from this intracellular pathogen on its surface. So this is some antigen, some molecule from the surface of this intracellular pathogen. It's going to display it here where other immune cells can detect it. So we're just going to follow this top pathway here. So here you have it, one of your T cells. Your T cell, what a cytolytic T cell does is it goes around checking all your other cells for infection. It checks to see if there's anything in this receptor here. We'll go over this in a lot more detail when we get to immunity. So this T cell is going to check. It's going to say, oh, is this receptor empty or is it full? If it's full, this T cell now knows, oh, this cell is infected. There's a tricky little parasite hiding in there. So what this T cell is going to do is it's going to kill the infected cell. So it's going to kill it, pop it open, kill it. Once it pops it open, this parasite will now be exposed and the rest of the immune system can come and handle it. So again, this is an act of self-sacrifice. This cell has not killed itself. It has been unable to kill itself. But what it does is it flags down this other cytotoxic T cell or cytolytic T cell, which can kill it, which can do the job for it. And again, it's an act of self-sacrifice to prevent the spread of infection. The last item on self-damage is excessive immune response. So inflammation response is a positive immune response in most cases because it helps our bodies transport immune cells and oxygen to a site of infection. But some pathogens trigger, and also fever. Fever helps us fight pathogens because it brings our body temperature above the temperature tolerance of the pathogen so that the pathogen is weakened by the heat, and that helps our immune cells deal with the infection more efficiently. But some pathogens trigger an overreactive immune response that can, so much inflammation, so much fever and inflammation that's so severe that it can cause organ damage and shock and death. So shock is a sudden and life-threatening drop in blood pressure. So when you have an excessive inflammation response, it can cause organ damage. Also, inflammation is the dilation of your blood vessels. When your blood vessels dilate or widen, it drops your blood pressure. If your blood pressure drops too far, it can be life-threatening because you're not getting enough oxygen to your brain. When your fever, I should add to this that if your fever gets too high, your fever can get, so the purpose of fever is to bring your body temperature up above the temperature tolerance of the pathogen. But if your fever gets too high, it'll bring your body temperature up above your own temperature tolerance. That's going to cause your own enzymes, your own proteins to denature. What you're trying to do is denature the proteins and melt the plasma membrane of the pathogen. But if your fever gets too high, you're going to denature your own proteins.